All right, I'm Josh Wagner, and today I'll be going over protein metabolism and how our body breaks down protein and stores that. So essentially, what is protein? Protein can be found in the foods that we eat, and it's made up of amino acids. So basically, you have complete proteins and you have incomplete proteins. So complete proteins are going to be proteins that include an adequate amount of all the necessary amino acids, whereas incomplete proteins are going to be proteins that only include one of those amino acids or a sufficient amount of a couple, but not all of them. So basically, there's 10 essential amino acids and 10 non-essential. The 10 essential can be remembered with the acronym Private Tim Hall. So phenylalanine, valine, and threonine are the PVT, and then tryptophan, isoleucine, and methionine are the TIM, and then for the HALL, histidine, arginine, leucine, and lysine make up that. And leucine and lysine are important because those two are known as ketogenic. So basically with a steak, this would be more of a complete protein that has a lot of the necessary, adequate amounts of the necessary amino acids. So this is going to be mechanically digested in the mouth and then pass through the esophagus until it hits the stomach. So starting in the stomach, the chemical digestion is going to begin as pepsinogen and HCl form pepsin, which is going to start to cut the polypeptide chains in the stomach and pass them into the duodenum. So next in the duodenum, there's 3 important enzymes here that we need to take note of. And these are kind of scissor-like enzymes that work to cut the polypeptide chains in the duodenum. So these are trypsin, carboxypeptidase, and chymotrypsin. And you can see these 3 here are working to cut those polypeptides in the duodenum and pass them into the jejunum, where the brush border and the pumps at the brush border become very important in the jejunum. So at the jejunum, the sponge effect takes place, and basically the Na plus and the K plus pump is first. And this is important because this sets the ion gradient and sets up facilitated diffusion. So without this pump going first, the other pumps aren't going to be able to act sequentially. As you can see, H plus being pumped out here as di and tripeptides are being pumped in with Na plus, and then H plus comes in with Na plus again and amino acids, where the polypeptides are cut and released into the blood vessels. So after the blood vessels, it's going to move to the liver. As you might notice, I have the liver highlighted here, and we're going to move to the back and take a closer look at that. So within the liver, there's 2 processes that take place as the amino acids enter. The first of those 2, and this happens 85 to 90% of the time, is known as transamination. It's basically known as the recycling of amino acids because the amino acid is broke and separated into 2 groups. These 2 groups are known as the carbon skeleton and the amine group. And the way in which this is broke is transaminase and vitamin B6 work to split it apart. So this part over here, the carbon skeleton, is used for energy. So this is like glycogen, glucose, acetyl-CoA, triglycerides. They're used for the immediate body energy, whereas the amine group is more of the stored glutamate. So this is used to make new amino acids such as muscles, hormones, or neurotransmitters. Again, this is going to happen 85 to 90% of the time in healthy individuals that undergo a normal protein metabolism. So basically, PLP and H plus form to make ammonium, which is really toxic. So this ammonium undergoes the urea cycle, where it's converted to urea, and H2O is added, and then it is sent to the kidney. At the kidney, the urea is turned into urine and is released from the body at that point. Now, the second of these 2 processes is known as oxidative deamination. In healthy individuals, this only happens 10 to 15% of the time and is usually when we're at a state of exercise. This is for the body to obtain fuel. So basically, it uses glutamate dehydrogenase. And essentially, if your body is undergoing this process too much or more than 15% of the time, I'll say you're doing this 40% of the time, your body can get leaky NH3 and NH4, which can cross the blood-brain barrier and be very toxic to the brain. Also, if this process goes wrong, it can increase your levels of anxiety and have a tougher feeling of relaxation. So essentially, I'll show you what this looks like up here. So in the normal levels of protein consumption, we're getting a normal level of stimulation within the brain. Whereas if we have excess, you can see our excitatory is way up here with anxiety going up and feelings of relaxation being decreased. After that, I hope that everyone here that watched this video can learn to make good eating decisions so that their mental health can improve and their gut health will stay in check. Thank you for watching.